project is tiger detection and individual identification. So, the contents include introduction, need of tiger senses, design, phases and explanation, advantages, disadvantages, conclusion and reference. Looking into the introduction, we can simulate human visual system by using computer vision. Adding machine learning to it will give a very much accuracy than a human visual system. Why do we need tiger senses? We know that India is the home of 70% of global tiger population. That is 70% of the tiger resides in India, which has tremendously reduced in that century. Tiger has been ranked as the 9th species that is undergoing extinction by the IUCN. Being our national animal and we call us umbrella species, it is the topmost animal in the food chain, and anything that affects the tiger will also affect other animals in the other levels of food chain. So it is our duty to protect tiger. Looking into the design, this is a symbol design that is consisting of 4 phases and a symbol graphical user interface. Phase 1 is tiger detection. Here the images consisting of tiger is only detected. Phase 2 is extraction of interested region. Here the interested region is flank area of the tiger and it is extracted from the whole image. Phase 3 is mapping extracted regions into monochrome. Here the extracted flank regions are converted into monochrome formats. Phase 4 is clustering for individual identification. So the clustering is done as agglomerative clustering is done to identify individual tigers. As we can see, the data flow diagram consists of an input which containing a folder of random images which we have obtained from camera traps, and this is detected and the images of only tigers are outputted from this detection phase, and these images of tiger is an input to the flank extraction phase where only the flank will be extracted and outputted. Then this will be given to the monochrome conversion phase where this is converted into a monochrome format where the binaries are only available, and it gives input to clustering phase. Looking in deep to each phase, phase 1 is detection. As I said, the input will be the folder containing of random images from the camera traps, and the output will be the images of only tigers to a new folder. The technology used will be object detection model that will be trained to classify images as 2 classes, mainly the tiger class and non-tiger class. Only the tiger class will be given as the output. It is implemented using TensorFlow Keras deep learning library with the use of Google Teachable Machines. So the datasets used here are collected from Amur Tiger Re-Identification Challenge Test and trained among with annotations. Phase 2 is the flank extraction phase where the input will be the output of the previous phase, which is a detection phase, and the output will be consisting of only the cropped images of interested region, which is the flank. The technology used here is symbol cropping technology using custom design cropping tools. It is implemented using Pygame, a game development library, and various file automation tools.
Now we have the folder called mono, which contains the image of the flank region. Here we are converting this to monochrome. That is, just images are mapping to binary. This process is recommended for better accuracy. This process can be implemented using Python imaging library and NumPy. In the fourth and final phase, monochromatic image from the last phase is used as an input to the new phase. Agglomerative clustering is used for the individual identification of tigers. We have taken distance threshold as a termination condition for this agglomerative clustering. The output folder represents the individual tigers, that is the clustered result. We have identified the individual tigers through all this process. We have designed our graphical user interface using tkinter. Directory picker is used for selecting the directories. Here we can see the interface window. Initially, folder containing random camera traps images are browsed using those browse button. The launch button is used for selecting the flank region, that is done manually. Convert button is used for the conversion of those flank regions to monochromatic images. By clicking the identification button, the clustering process starts. The output will be given by given as individual tigers or individual clusters. Verified out.dar represents the result of the whole process. Here we can also see the individual sets of folders of individual tigers. The classical census approach contains a huge risk factor. It is mainly reduced here. Our project provides better accuracy and less time consumption than the classical approach. It will also reduce the human intrusion, and thus we can provide or prevent other illegal activities like poaching in the forest, etc. Now let us analyze the disadvantages of our project. The accuracy of our project is highly concentrated on the camera quality that is used for camera traps. Manual error in cropping might also affect the result. And when we conclude, our project meets lots of our expectations and objectives. It have also a scope to grow further. That is, we can eliminate the manual cropping process by further improving the postnet. The current annual postnet is not well accurate, so we couldn't include it in our project. These are our references, and thank you.
Now to a quick demo of our project. As we can see, this is the program, and we have a folder containing random images which is named as tset. It contains some random images. We will be using it to showcase the detection feature of our project. As we can see, there are some random images. Some of them are tigers and some of them are not. So in this phase, we will be taking this data and will be generating a folder containing only the images of tigers. So when we launch the program, then an interface pops up, and this is made from tkinter. So we have to select a folder. For that, we have to click the browse button. Then we have to select the random folders, the folders containing random images of different animals. Okay, it's not responding. It happens sometimes because my PC is a low spec PC. So as we can see, the tiger data folder is empty. Now it's starting to run. Okay, it shows the path and the status, which means how many images it processed. And now we can see the tiger data folder has 4 images and all of them are tigers. Okay. Now we have to extract the flank region in order to identify the tiger. We need to individually identify the tiger. So we have provided a tool which can extract flank. For this image there is no flank region, so we can simply ignore this one by clicking the terminate button. Then this is the proper image of a tiger where we can crop the image, crop the flank region. This is another one where we can simply crop the flank region. We are expecting to get multiple images of a single tiger from the dataset. That's why we have an option to ignore a particular photo if the flank is not extractable. So we can see in the mono folder we have all the extracted flank regions. Now if we launch, then we have to convert it to monochrome. It's recommended for better accuracy. So it's a simple process. We will be just mapping it to binary. So in the monochrome folder we can see the mapped images. So then we will be clicking the identify, which will be used to identify individual tigers, which it's basically agglomerative clustering. So when we click identify, it will run a program and it will calculate the feature vectors for each images and cluster it. So we can see verify out dir clusters 3. So 3 clusters are formed, which means there are 3 individual tigers in the data provided. So each cluster contains the images of individual tigers. So that's it. This is the GUI and this is the overall process. So let's look at the program. Okay. So we have used tkinter for the interface, then TensorFlow for the TensorFlow Keras as the deep learning library, Shuttle, OS, Globe, etc. are used for file automations, and sklearn is used for importing the clustering feature, and Pygame is used as a tool for cropping the image. So that's all. Thank you.